Welcome back, everyone, to the Emerging Topics in Biomolecular Magnetic Resonance series. Just a short reminder, as always, that we go by the Gordon Research Conference policy of no private recordings, and this is entirely unnecessary today since we'll be recording the lectures. And for the Q&A part at the end, please use the Q&A, which you can find at the bottom to the right of your screen. And you can also raise your hand to speak if you'd like to have a more interactive Q&A session. Otherwise, we'll read out the questions from the Q&A box. So today, once again, we have two very exciting talks lined up. First, we have Markus Weingarth and then Martin Blackledge. And Martin Weingarth, a short introduction, did his PhD at EPFL with Jeffrey Bodenhausen. He then went on to Utrecht with Baldus as a FEBS fellow. And after being a group leader there for some time, he's now a professor in Utrecht. And his work spans solid state NMR proton detection, method development, and membrane proteins. And today, I think he's going to tell us about antibiotics and cell membranes. So Markus, the stage is yours. Let me just...
Thank you. Thank you very much for the kind introduction, Darren. I would like to thank the organizers also very much for having me today. I think it's a great idea to organize these webinars to exchange with each other in these strange days. And I hope that everybody out there is well and safe. Can you please confirm that you see my slides?
Not yet.
Not yet.
Yeah,
no?
perfect.
Great. All right, so on with science, let me give you a crisp introduction on our research. So our research is focused on antibiotics. We are interested in the mode of action and the mechanism of antibiotics. And once upon a time, less than a year ago, antimicrobial resistance was considered as the biggest threat for human health. And according to a recent study commissioned by the British government, antimicrobial resistance would be the killer #1 in the near future, causing more deaths than cancer. And meanwhile, we live in a different world, the world of COVID-19 that had eclipsed everything. But antimicrobial resistance is not going to disappear miraculously. On the contrary, COVID-19 will presumably lead to an increase in antimicrobial resistance due to the heavy use of antibiotics to treat COVID-19 patients. If you think of the infamous hydroxychloroquine that you may know from Trump, that was always administered with the antibiotic azithromycin. So in order to face the challenge of antimicrobial resistance, we urgently need novel antibiotics, and we especially need antibiotics that are refractory, that are robust to antimicrobial resistance. And that's the reason why we work on lipid-2 binding antibiotics. So what's that? Our target, lipid-2. The target, lipid-2, is the precursor of the bacterial peptidoglycan network, and peptidoglycan forms a protective essential hull around bacteria they die without. Peptidoglycan is composed of repetitive sugars that you see here in blue that are connected via short peptide bridges. Peptidoglycan is in constant turnover and is maintained and constructed via its precursor lipid-2. Lipid-2 carries one sugar peptide, peptidoglycan subunit, that is attached to a unique pyrophosphate group that is connected to a complex lipid. It is assembled here in the cytoplasm, then flipped across the plasma membrane, where the head group is chopped off and integrated into peptidoglycan, and the rest is cycled back for the next round. And this lipid-2 cycle is essential for bacteria, and any drug that interferes with it is an antibiotic. And this notion was famously validated by vancomycin. Vancomycin used to be an antibiotic of last resort, and it binds here as a lipid-2 pentapeptide. But you all know that peptides can be mutated, and that's why many bacteria are resistant to vancomycin these days. So we work on antibiotics that have a smarter binding mode. They bind here at the pyrophosphate. And since phosphates cannot be mutated, this binding mode is ultra-refractory, ultra-robust to the development of antimicrobial resistance. And it's also very powerful. For example, a few years ago, tioxibactin was discovered, which is an antibiotic with extraordinary activity, and it also binds lipid-2. And I will show you later our research on tioxibactin. In general, these lipid-2 binding antibiotics come in two flavors. They can either use lipid-2 as an anchor to form deadly pores across the plasma membrane, and here the best example is niacin, or they can simply tightly bind lipid-2, blocks the lipid-2 cycle, and this is fully sufficient to kill multi-drug resistant bacteria effectively. However, these binding modes are poorly understood, and there are hardly any structural data available and no data at all in membranes. And this paucity of structural data is a major limitation for the use of these powerful compounds to develop antibiotics. It is our ambition to change this. We want to understand these binding modes at high resolution and in natural membranes, and ideally in physiologically relevant cell membranes. And I'd like to give you one catchy example why we need to understand membrane active drugs in membranes. So this here is niacin. It's a poster child of the lipid-2 binders, and there are around 100 papers every year on niacin. So niacin features 5 signature thyroether rings that adapt A, B, C, D, E, and rings A and B target the pyrophosphate of lipid-2. And there's already a beautiful structure available of the pyrophosphate lipid-2 interaction that was solved by our collaborator Evian Berking with solution NMR and DMSO. But you all know that things like micelles or DMSO are not ideal membrane mimics, and that's why we wanted to revisit the structure. And we have here the original HSQC spectrum from the NSNB paper that shows niacin bound to lipid-2 in DMSO. So let's see what happens if we measure exactly the same complex in natural membranes. And you see that's a difference like day and night. The spectra are completely different. And this must mean that the complex is also completely different in natural membranes. And this is something that we published 2 years ago. And the first author of the study, John Medeiros Silver, will soon join Mei Hong's group at MIT. Meanwhile, we have substantially progressed with the structure definition, but today I'm not going to talk about niacin, but I'm really excited to show you our latest work on tioxibactin. So what is tioxibactin? Tioxibactin is an antibiotic with outstanding activity against a broad range of clinically relevant pathogenic bacteria. Its discovery in 2015 made a tremendous splash and was globally heralded as one of the drug's game changer in the combat against pathogenic bacteria. It was considered the first novel antibiotic for more than 30 years, and the paper has been cited around 2,000 times. Tioxibactin has a real perspective. It is pretty far in clinical trials. So what is tioxibactin precisely? It's an 11 residue peptide that features lots of unnatural amino acids, including 4 D amino acids, and there's a ring motif at the C terminus. Here in the ring, we have a very funky, very unusual amino acid that is called enduracidine. And before we started with tioxibactin, a lot was already known. So it was known that this ring motif at the C terminus targets a pyrophosphate. It was also known that somehow the N terminus is essential. If you chop it off, the activity is gone. And it was also strongly suggested that the N terminus adopts beta strand conformation in the bound state. This was shown in a very beautiful publication by the group of Lewandowski in mice cells. Furthermore, it was also known that the sequence of D and L amino acids cannot be changed. If you change it, the activity is gone. In a similar way, and if you change the order of polar and apolar residues, you also lose all the activity. So a lot was already known before we started, but there were no data in membranes, and that's what we wanted to contribute. And therefore, we teamed up with Ishwa Singh from Liverpool University. Ishwa is a leading expert in synthetic tioxibactins. In particular, Ishwa discovered that this funky residue here, this enduracidine, can be replaced by conventional hydrophobic amino acids. This does not reduce the activity, but it reduces the cost of synthesis by 3 to 4 orders of magnitude. And Ishwa also exchanged this glutamine here for an arginine to improve the solubility. So we worked on these improved tioxibactin analogs, and in order to make them NMR amenable, we introduced 6 carbon-13 labeled residues that covered the entire molecule, including the N terminus and this ring motif. And this is the first solid state NMR-HSQC spectrum of tioxibactin bound to lipid-2 in the UPC liposomes. It's a simple spectrum. We count 1, 2, 3, 4, 5, 6 signals that relate to the 6 residues, and it's strongly indicative of complex formation because it looks totally different than the free state. And eventually, using phosphorus solid state NMR, we can unambiguously confirm that we look at the bound state. This is a phosphorus solid state NMR spectrum of liposomes doped with lipid-2. The big signal relates to the bulk lipids, and the 2 smallest signal to the pyrophosphate of lipid-2. And once we add tioxibactin, then the pyrophosphate signals drastically shift, which confirms complex formation and means that our samples are okay. Next, we looked at the secondary structure of the bound drug, and therefore we acquired this beautifully resolved 2D carbon-carbon spectrum that we can readily annotate all the signals. And then from these chemical shifts, you all know this, we can readily determine the secondary structure using so-called secondary chemical shifts, where positive values mean alpha helix and negative values mean beta strand. And it's clear from this data that the N terminus adopts beta strand conformation. Next, we're interested in the supramolecular arrangement of tioxibactin and the complex. Therefore, we acquired 2D carbon-carbon spectra with short and long mixing time. With the short mixing time, we only get intraradical contacts, and with the long mixing time, we get interradical contacts. You see many more contacts here with a long mixing time, and we're going to superimpose now these signals in the green boxes. And please focus on the signals with red labels. These are the interesting signals. These are long-range contacts of many signals. For instance, we have here long-range contacts between residue 2 and 6, between residues 3 and 7, and between residues 2 and 7. And if we consider that the backbone distance between residues I and I+2 in a beta strand is 6 angstrom, this would mean that the distance between residues 2 and 7 is around 15 angstrom. And you all know this is not possible between two carbons. And this must mean that these contacts cannot be intramolecular, they must be intermolecular, which can only be explained by antiparallel beta strands. We could unambiguously explain that tioxibactin and lipid 2 oligomerize, therefore we use fluorescence spectroscopy and pyrene-type lipid 2. Once two pyrene molecules come close to each other, they give a unique so-called aximer signal at 490 nanometer. This is exactly what happened when we added tioxibactin to liposomes that were doped with pyrene-type lipid 2. You see here the buildup of this unique signal at 490 nanometer. So these data clearly demonstrate that tioxibactin and lipid 2 oligomerize. And potentially they can oligomerize to something really big. And that's what we were interested in. So we sought to detect these oligomers with fluorescence microscopy. Therefore, we used NBD lipid 2. You have here the distribution of this TAC lipid 2 on the surface of giant unilaminar vesicles. And indeed, once we added tioxibactin, we observed these massive micrometer scale clusters. And that was a really surprising, a really remarkable finding. And while we don't have any proof, we do believe that these clusters mark an additional killing mechanism. And we believe that cluster formation extracts lipid 2 from the cell wall synthesis cells. And that cluster formation and killing are related is plausible once we look at the timeline of cluster formation. The initial binding of tioxibactin to lipid 2 is fast, and cluster formation requires a few hours, and killing also requires a few hours. If this is a coincidence or a functional correlation, that's something that we need to see in the future. All right, so now we know that tioxibactin and lipid 2 form these massive clusters, so let's look into these clusters at high resolution and let's determine the interface at high resolution. The first thing that we wanted to confirm was if this C-terminal ring motif really contacts the pyrophosphate is also really strongly suggested by a large body of literature. And therefore we acquired a 2D proton phosphorus spectrum where we transferred magnetization from the amino proton to the pyrophosphate. And this spectrum was acquired with a precious help of Marino Lelli at CHEM and was financially supported by INEX. So we have here the dimension of the amino protons. At the phosphorus dimension, you see that only the pyrophosphate, but not the bulk lipids, talk to the amino protons. Next, we try to annotate these signals. Therefore, we compare this proton phosphorus spectrum to a proton nitrogen spectrum. Now we need to realize that this proton dimension and that proton dimension are the same, and then we can transfer the proton assignments from here to there. And you see that these signals match spot on with residues 9 and 10 of the ring motif. So thereby we establish directly that the ring motif coordinates the pyrophosphate. What we also need to understand is the role of the lipid 2 head group in the complex, so the role of the sugars in the pentapeptide. Especially the role of the pentapeptide is critical because it's there where bacteria introduce modifications that are known to cause antimicrobial resistance. To make the lipid 2 head group NMR amenable, we needed to carbon-13 label it, and that was a very hard nut to crack. But thanks to the heroic help of Evian and Zhao, it worked out. So we have here a 2D carbon-carbon spectrum of lipid 2. These are the sugars, and this is the pentapeptide region. And I will briefly show you on the example of niacin how we can use this to determine the interface. We have here an overlay of 2D carbon-carbon spectra of the niacin lipid 2 complex. In magenta, only niacin is labeled in the complex, and in green, both niacin and lipid 2 are labeled in the complex. And many of these green contacts without magenta correspondence provide us with precious information on the interface that we can use for an NMR structure determination. So let's use this approach to determine the interface between tioxibactin and lipid 2. These are 2D carbon-carbon spectra that are very well resolved and provide us with clean information on the tioxibactin lipid 2 complex. Here we have added all the interfacial contacts in the primary structure of tioxibactin. You see that the interface is indeed at the ring motif here at residues 9 and 10. And we see that almost all interfacial contacts relate to the Myrnax sugar that is directly attached to the pyrophosphate. There are hardly any contacts for the glucanac sugar that must be distal from the interface. The big surprise was that we did not observe any contacts for the pentapeptide. And this is a dipolar-based spectrum. This means, or this strongly suggests, that the pentapeptide is highly mobile in the complex. And indeed, we could readily confirm this with the so-called TOPSy spectrum, where we only detect scalar residues, or where we use scalar transfer and only detect highly mobile residues. And indeed, here we only see the pentapeptide. These are residues 5 and 4, residues 3, and residue 2 of the pentapeptide. So the pentapeptide is highly mobile in the complex. Let's put this all together. Thus far, we have learned that the ring motif coordinates the pyrophosphate, the Myrnax is directly at the interface, glucanac is distal from the interface, and importantly, the pentapeptide is highly mobile in the complex. And these data beautifully explain why tioxibactin has such a gorgeous activity against a broad range of bacteria and why it is so robust to the development of antimicrobial resistance, because tioxibactin does not seem to care about all the modifications that bacteria introduce in the pentapeptide. So the last thing that we needed to understand in order to perform a structure determination was the membrane topology, and therefore we used an old trick developed in the lab of my home. It's a so-called mobility edited or G2 edited experiment where we transfer magnetization from mobile water and mobile lipids to tioxibactin, and you see that tioxibactin intensely interacts with both water and lipids, and that's typical for a compound that sits at the water membrane interface. The only exceptions are here, and these are the long hydrophobic side chains, isoleucine 2 and isoleucine 6. They only talk with the membrane because they partition into the membrane, and thereby we have established the membrane topology. So with all this information on the structure of lipid 2, the structure of tioxibactin, the structure of the interface, and the topological data, we could perform a structure determination in Haddock, and therefore we got prizes from this charming gentleman. This is Alexandre Brevin, the lead developer of Haddock. And this structure determination worked out pretty well. We could determine the complex with pretty high accuracy, with an RMSD of less than 2 angstrom on tioxibactin in the complex. In yellow, we have here the antiparallel tioxibactin fiber, and you see that the ring motif and the cationic C-terminus of tioxibactin tightly coordinate the pyrophosphate. The rest of the interface is loosely defined, and this fuzziness of the rest of the interface enables tioxibactin to target other cellular precursors. Tioxibactin also targets cellular precursors that are called lipid 1 and lipid 3 that all have the pyrophosphate in common, but slightly different head groups. And this is another mechanism how tioxibactin evades resistance development. What I really liked about the complex structure was that it explains this distinct sequence of DNL amino acids. And you see here a clear-cut separation of hydrophilic and hydrophobic residues. Hydrophilic ones face the water, hydrophobic ones face the membrane. And this is only possible with this sequence of DNL amino acids. If you imagine it a hypothetical all-L tioxibactin, here you would not have a favorable distribution of hydrophilic and hydrophobic residues. Some hydrophilic ones would face the membrane, and some hydrophobic ones would face the water. So a favorable distribution really requires a stereochemistry, and I know what you're thinking, that this is just totally obvious, and I admit in hindsight it is pretty obvious, but nobody thought about this before. All right, so I hope thus far it was a relatively consistent story, and I wish that we had stopped our project at this moment, but unfortunately we also had data on the membrane influence, and that's when trouble started. You see here an overlay of spectra of tioxibactin bound to lipid 2 in blue in switterionic membranes and in red in anionic membranes. And you see many changes in anionic membranes here, there, and also there. So there's a global impact of anionic lipids on the binding mode of tioxibactin. And we wondered if this global impact of anionic lipids was something real, or if this was something due to our synthetic liposomes. And in order to understand if this really happens in cell membranes, we acquired data in cell membranes. These data were acquired in the cell membrane of Micrococcus flavus, and indeed, the signal changes here, there, and especially here are virtually the same. You see this conflation is really the same. This tells us that the impact of anionic lipids is of physiological relevance. What was really strange was that the sensitivity dropped by almost an order of magnitude in anionic membranes. And if you're a solid state spectroscopist, this immediately triggers you and you think about dynamics. In the solid state, there can be a relation between mobility and sensitivity. So there can be an inverse relation: the higher the mobility, the lower the sensitivity. And indeed, that was exactly what we found using relaxation measurements. We acquired T1 row data, and these data unambiguously demonstrate that the bound state of tioxibactam is much, much more mobile in anionic lipids. And this triggered us, and we measured T1 row relaxation data in a number of different membrane environments, and there was a clear correlation. The more negative the membrane environment, the higher the mobility. And this leads now to the elephant in the room. What does this mean for the binding affinity? So we quantified the binding affinity using ITC, which is the gold standard to measure binding affinity, and we acquired ITC data in SWIT ionic membranes and in anionic membranes, and the outcome was very clear. Binding affinity is indeed much lower in anionic membranes. The binding constant is increased by 2 orders of magnitude. So we end up with a clear-cut inverse correlation between dynamics and affinity. The higher the mobility, the lower the affinity. And I want to give all the credit for these ITC measurements to my student Rhythm Shukla, who did really a fantastic job in acquiring all these data. This was not easy because we had to work with an older ITC machine in another department. We have meanwhile our own brand new ITC machine, which is the big boost to our research. So let's try to put this all together. We have sourced the structure of the complex and natural membranes. From this structure, we can learn a lot on the pharmacophore, and we see that tioxibactam combines specific tight binding to the pyrophosphate and fuzzy binding to the rest of the interface. This enables tioxibactam to maximize its number of targets and to effectively evade resistance development. We have also seen that anionic lipids have a drastic influence on the binding modes. The binding affinity is drastically reduced in anionic lipids, and that's something really surprising because all bacterial membranes or virtually all bacterial membranes are anionic. So how come tioxibactam is such a wonder drug if its affinity is actually pretty bad in anionic membranes? We don't have an answer for this, but we can speculate, and we speculate that the dominant killing mechanism is actually this cluster formation here, because cluster formation does not depend on the membrane charge, and the timeline of clustering and the timeline of killing coincide. But we don't have a proof for this. So this brings me to the acknowledgements. The key players in this work were my PhD student Rhythm, my former PhD student Drau. I also would like to thank Ishra for synthesizing all these gorgeous tioxibactam analogs. All these lipid 2 synthesis efforts were done together with Evian, and a special thanks for Marino for helping us out with the phosphorus measurements. I'd like to thank you all for listening, and I'm happy to take any question. Thank you.
Thanks for the fantastic talk, Markus. While the questions are coming in, I'll ask a quick one. For the phosphorus interaction that you measured, was that CP spectra?
Yeah, brute force. Brute force.
And then also the age-old question of whether you need lots of imprecise distances or a few precise distances. Maybe you could comment on if you think you could refine more, or if this would help in your structures.
Yeah, it would certainly help. It would certainly help to have more precise distances. I think it's also clear from my presentation that we were blind on 5 residues, so only 6 residues were labeled. So for 5 residues, we do not have direct experimental information, and it would certainly help to have a couple of more restraints to increase the definition. Yeah.
Thanks. And then Christian just messaged that he would like to ask something.
Okay, thank you very much. So I have 2 questions. One was, I mean, you showed that isoleucine 2 and 6 were inserted in the membrane, but there was another phenylalanine and isoleucine 5 as well, and they were also in the final structure pointing in. So what about them? And the other thing regarding the mechanism of killing, I did not really understand why it needs the polymerization or the aggregation of the peptide to do the job. I mean, if it just targets one, irrespective of the others, so I was wondering how that flows out from the start. I mean, maybe it's unrelated, but I was interested in your view on that.
Thank you. Great question. So the first question is relatively straightforward to answer because residues 1 and residues 5 were D amino acids, and we could not afford to label them, so they were not labeled in our Okay. We're working on this. We're working on this. And the second question is fantastic. I do not have a perfect answer for this. So we have discussed about the killing mechanism at length also with industry. Meanwhile, what might be a little bit misleading from our ITC data is that ITC data might not be at equilibrium. So you do not know K on and K off in this case. Therefore, you would have to do special ITC measurements, which is not our expertise. We're also working on this. So it might be that we're actually not in equilibrium when we acquire these ITC data. But this is speculation. This is something that we do not know.
Then following up on that, would it be conceivable that that you see this aggregation only because you work maybe at unphysiologically high concentrations with the compound?
It oligomerises at low concentrations, at very low concentrations. This is quite known in the field, and this is also something that we prove with our fluorescence measurements that we do at very low concentrations. We did not investigate these massive complexes, so these microscale complexes at lower concentrations. So I cannot rule out that they do not form at physiological concentrations, but it oligomerises at low concentrations. Thank you.
Okay, then we also have some questions in the Q&A. So Alon's Lens would like to know, what was the lipid to protein ratio in your samples, and would you look at would different ratios somehow affect the quality of the proton detected spectra?
It's an excellent question. Thank you very much. The lipid to protein ratio was 1 to 100. And if we went for lower concentrations, it would certainly affect our quality because we would have much less sensitivity. But for the cellular spectra, we did the cellular spectra with a lipid to protein ratio of 1 to 1,000. So, and you get virtually the same spectrum. So it's just a numbers game. If we have a lower protein to lipid ratio, then we just need to acquire for a little bit longer.
Then Ashish Arora asks, this scales in the two fluorescence micrographs are different. This means that the GUV showing clustering of lipid 2 is smaller in size. I guess it's more of a statement, but
I honestly do not understand the question. I'm sorry.
Okay, so if you'd like to raise your hand and clarify, please do. In the meantime, I'll go on to let's see if I can unmute you. 1 minute. Okay.
Hello.
Yeah. Yeah, we can hear you.
Yeah, hi. So Markus, if you just go to that figure where you have shown these two, the GUVs.
Yeah, it's a pleasure. Just a second, please.
I was surprised by it actually.
Okay. Positively or negatively?
No, see, the scales are different, and the circles are exactly the same size, but that 20 micron bar is different. So I was just wondering why that has been chosen to represent this result.
We presented the circles so that they had the same size. This just
yes, but that means that the vesicles have different size.
Well, in one sample there are thousands of vesicles, and you just pick some that look nice, and
okay,
yeah.
So you uniformly see this clustering in many vesicles that are sticking together.
Also, we would also see this, yeah.
Okay, yeah, that's all. Thank you. Yes.
Okay, thank you for your question. Thank you.
Okay, then we'll go to Ashish Arora, who asks, Teixobactin is bacteriostatic and not bactericidal. How can your experiments explain this?
It's a very good question. Thank you very much. It's a very good question. Thank you very much. Cluster formation would, for example, explain it, because cluster formation is a lengthy process that requires hours, and that would represent, and that would you would see this as a bacteriostatic effect in your experiments. If you compare this with niacin forms deadly pores or pokes deadly pores in the membrane that kill your bacterium immediately, and that would be a bactericidal effect. I hope that's okay as an answer, but I wouldn't have a better one, I'm afraid.
Thanks. Then James Gibson says, when you say that the complex from Haddock had an RMSD of less than 2 angstroms, to what are you comparing the computational structure? So are you comparing to another structure, or is it the cluster, I guess?
It's a cluster. You compare to the average structure.
Mm-hmm. And then we have a question about the rotor size during magic angle spinning and the rate that you use.
It's always at 60 kilohertz, and we use 1.3 millimeter rotors because that's the only rotor size that we have available at the moment for proton detection. I wish we had smaller rotors, but we don't have that.
Thanks. And we also have another, well, I see the question disappeared, but there was one about lipid 3 from Aurora also, which I found interesting. So the question was, I guess, if you would see binding to lipid 3.
I would assume so. That's what you see in the literature. We don't have lipid 3 available. It's quite difficult. It's quite difficult to get synthetically. Yeah.
Thanks. Let's see. There's just a couple left. So Anonymous asks, how big are the oligomers, i.e., how many monomers?
Don't know.
Okay. And then the final one was cholesterol ever used during the preparation of the membrane mimetics?
No.
DOPC cholesterol.
No, we haven't tried this out. It's an interesting question. Why I cannot, of course, I cannot ask back. No, we haven't tried this out. I need to think about this, why we should.
Well, maybe we can then do that in the more relaxed session at the end. And
sure.
Then I'll turn over the platform to Christian for the next one.
Thank you very much.
Thank you. Thanks again for a great talk.
Okay, now we switch over to the second talk by Martin Blackledge. Let me just introduce him a little bit. He did his PhD with George Rada in Oxford, 1989, and there he was kind of disappointed with moving objects and localized spectroscopy, and rather than chose moving objects in as molecules, i.e. studying dynamics with of in vitro prepared molecules. And I mean, this is really the topic of the work that he had then during the time in Zurich, where we had the pleasure, I think, of overlap of half a year or something like that. Martin started working there on relaxation. I think at the time it was more or less exclusively on protons, if I'm correct. And this, of course, is still a hot topic. And but then founded his own group, and then worked on many different topics. I mean, really characterizing intrinsically disordered proteins, but also folded proteins with RDCs, PREs, ZECs, but also contributing a lot to methods actually to characterize this. So he's really an expert both in NMR parameters, really developing them, but also then interpreting or translating them into ensemble description of these intrinsically disordered proteins. And of course, phase separation, which is one of the big topics now from intrinsically disordered proteins, is his interest. He also is interested in not only mobility in liquid state, but also in solid state. I should mention with the flexible mechano, of course, this was a big step. Actually, I would say that one really can start describing these ensembles and tie in the parameters that are measured, like RDCs and so on, as I was mentioning. So I'm looking forward to your talk, Martin. You said that half of it is unpublished work or work that you have not presented in the past. So really looking forward. The floor is yours. The virtual floor is yours.
Martin, you should unmute yourself.
Yeah.
Sorry, I'll mute myself. I thought I was
better now.
Sorry, yes. When you say that. Let's see. Can you see my screen? Not yet. Can you now? Oops. Sorry. Can you see my screen?
Yes.
Good, thank you. Okay, I will require no more answers. Okay, thank you, Christian. That was a really a very, very nice introduction. I'm going to be talking to you about, what NMR can tell you about how intrinsically disordered domains in viruses work. And you're probably sick of hearing about viruses, but for the next 22 minutes or so, you're going to hear about another 2 viruses. The first of these is measles, actually, which is not really a problem. As you're probably all aware, there's a vaccine for measles, and if only everybody became vaccinated, then there would really be no problem. But unfortunately, as you're probably aware, there are vaccine skeptics. There are people who don't take who don't get vaccinated. And of course, with the COVID-19 problem, as was pointed out in Science, I think 3 or 4 days ago, the coverage of vaccination is now likely not to be very complete and as a kind of indirect effect, but a potentially very dangerous effect. I used to have to explain what an R0 was, but I don't have to explain what R0 is anymore, I guess, because everybody knows. Suffice to say that measles is the most infectious pathogen that infects humans, and therefore that's why coverage has to be so high. Why are we interested in measles? Well, because first of all, it has a very, very simple proteome. And of the 6 or 7 proteins which are coded in its genome, 2 of them, which interact with each other, have very long intrinsically disordered domains associated with them, and therefore they're conserved, but they've escaped the scrutiny of structural biology because they're so disordered. These are the phosphoprotein and the nucleoprotein. The phosphoprotein is 75% disordered and the nucleoprotein 30% disordered. And they're involved in the replication complex, replication machinery. Phosphoprotein is about 500 amino acids long. It has a 300 amino acid intrinsically disordered N-terminal domain, a tetramerization domain, another disordered domain, and then a 3-helical bundle at the end. And this is known to form the polymerase complex, so it interacts with the polymerase, the viral polymerase, but also chaperones the nucleoprotein prior to capsid assembly. And the nucleoprotein has a folded domain of about 400 amino acids, and then a C-terminal intrinsically disordered domain, which is about 125 amino acids long. And what is known about the disordered P-tail is that the first 50 or so amino acids bind to the folded part of the nucleoprotein, and this keeps it in a monomeric state prior to encapsulation on the viral genome. And in fact, when you express the nucleoprotein in your favorite expression system, let's say E. coli, the first thing that happens when the protein is expressed is that it forms these long nucleocapsid-like objects by binding to any RNA that it finds in the cell, so any cellular RNA. And this, of course, gives you these long, beautiful spiral-shaped objects, which you can determine the structure of using cryo-EM. But because they're already assembled, you can't study the pre-assembly complexes nor the process of assembly. Nevertheless, when you do look at these structures, you can see how they're structured, how they're formed, and you can see that there's an arm. I hope you can see my cursor. There's an arm of one of the protomers which binds to the next protomer, and the arm of this protomer binds to the next one. And this is in fact exactly how the virus mimics this interaction with the phosphoprotein peptide. That's what keeps it in a monomer to make sure that this kind of thing doesn't happen. And inspired by this, we can also ensure that our protein no longer forms large oligomers by co-expressing it with a peptide of the phosphoprotein of different lengths, for example, and then we can study them in their pre-assembly complexes, or we can study assembly by adding RNA. And this is one of the things that we've done. These nucleocapsid particles, I should say that the intrinsically disordered domain of the nucleoprotein, this remains flexible, and this we were able to see by NMR measuring RDC's relaxation approximately 10 years ago by making these capsids in E. coli. So assembly really does work. You just add RNA to this monomeric chaperone, and the parts of the protein which were flexible and become rigid, their peaks disappear. The peptide that was bound, that is then leaves the complex, then the intensity of these peaks increases, so we can measure in real time the assembly process. In addition to that, we can form very nicely structured nucleocapsids, which are now bound to an RNA which we control, and then we can determine the structure of these, and this we did with our colleagues in the IBS in Grenoble. We can determine the structure using cryo-EM. We can identify key protein RNA interactions in the binding cleft between the nucleoprotein and the RNA. And we can make mutations, and we can see whether these mutations have any effect on assembly by looking at the assay which I just mentioned. And in fact, in this case, we find a key nucleic acid, amino acid, a nucleic acid interaction, which when we abrogate, there's no more assembly. But the reason one of the main reasons we wanted to actually do this, to isolate the pre-assembly complexes, was to examine really the role of this disorder. It's always conserved in these viruses, it must serve some purpose. So far we have no idea, except for the very end terminal end of p-tail, why it's all there. So the first thing we need to do, of course, is assign this. This is a 300 amino acid long p-tail. Suffice to say, it's essentially very, very disordered. There are some nascent helical regions, especially in the region which binds the nucleoprotein, but then two others which are about 100 and 200 amino acids downstream of this. The complex, this is a spectrum of the complex, so this is now a 92 kilodalton complex between N and P-tail. There are 450 disordered residues, all of which are assigned, so we can look at all of them. And we can study the complex either by co-purifying the N0P, so the NP complex, or by replacing the peptide that we've co-purified it with a longer p-tail, which has a higher affinity. Either way, the first thing we see, of course, is the interaction site that we know about. These are the first 50 amino acids. We knew about those. You can even co-crystallize this with the nucleoprotein. But there's another interaction site, which is about 150 amino acids downstream of this initial one. And this is obviously binding to the same protein. Now, we don't really know about the affinity of this, but we know this is tight, so this is nanomolar. So in order to determine the affinity of this, we need to make a construct which doesn't contain this one. And so this is what we did. I won't go through the details. You can see from relaxation measurements that this is kind of bipartite. There seems to be one region and then a dip and then another region. So we call these delta and alpha 4. We can measure relaxation dispersion at low stoichiometric ratios, and we can estimate the affinity, which is around 600 micromolar for this interaction, which is about 5 orders of magnitude weaker than the main interaction of these two proteins, of these two domains. It turns out that if you look carefully enough on other paramyxoviruses, like Nipah and other related viruses, then you can actually find similar sequences. The key sequence seems to be HELL. So you can imagine that caused a certain amount of wordplay in the lab. Because when you mutate HELL to AAAA, this retains the helical propensity, but the interaction has gone to, well, you can imagine, the interaction is not there anymore. More importantly, shall we say, using cell-based assays and changing the HEL sequence to 4 alanines, everything else in the mini genome assay seems to work. P still oligomerizes, it still binds to N via the main interaction site, but replication and transcription are abolished by this ultra-weak, the absence of this ultra-weak interaction. Now, bear in mind, if this is 5 orders of magnitude weaker, there are not many biophysical techniques which would allow you to detect an interaction which is 5 orders of magnitude weaker in the presence of another interaction, which is 5 orders of magnitude stronger. So I think this is a good advert for NMR. We can find out from NMR, of course, where it's binding on the nucleoprotein. It's actually binding on the distant region of the nucleoprotein, which is this variable region at the other end of the nucleoprotein. Of course, there are many, many degrees of freedom between this binding site and this one, so there's no difficulty of the second binding site reaching this. But of course, as it has an affinity which is very weak compared to this one, then it must be coming on and off very, very many times, and therefore maybe causing some kind of shield to the host immune system. We actually don't know why this interaction is essential. So this basically summarizes what I've just said. I'll just mention one other aspect, and that is that as this intrinsic, interaction is very, very weak, then obviously it will be a possible target for peptide-based inhibitors, because if we can't increase on 600 micromolar, then we're probably doing something wrong. So this is optimistic. The fact that we've now discovered this, I think, is something that we can work on. Now, this is yet another interaction. There are 4 or 5 different interactions between N and P, different domains of N and P. I'm putting this piece of information together with the fact that measles, N and P are known to co-localize in puncta in infected cells. And we wanted to know, and Serafima Guseva spent a lot of time trying to figure out whether mixing N and P actually forms phase separation. Turns out that it does. She could find out which elements of the 2 proteins are essential. That's the tetramerization domain and everything up to the C terminus, and the entire nucleoprotein. So those are the essential. That's the minimum system. She could elucidate the phase diagram and determine rotational correlation from time resolved fluorescence anisotropy, and look at overall dynamics, macroscopic dynamics using fluorescence recovery after photobleaching. As this is the minimum system which phase separates, we need to know something about the interactions between the 2 proteins. There are in fact then 2 between these 2 domains. One of them, it turns out, that we didn't know about before, that we discovered, that Serafima discovered. When this is abrogated, then the macroscopic dynamics of the droplets are somehow changed. This is so that the FRAP is somehow modulated by this. But the second interaction, this is the 4 here, between this helical region in the unfolded domain and this 3 helix bundle. When this is abrogated, then no more droplets are formed. And this we can abrogate with a single mutation. So this is obviously the key interaction, which is essential for the formation of the droplets. We don't get signal from these droplets. It's something we're doing in other studies that I won't talk about, but what we can see is the disappearance of NMR signal as we form the droplets, as droplets are formed, and as a function of stoichiometric ratio. And to cut a long story short, I won't go into much detail here, although we spent a lot of time looking at this. The data suggest that we need a 2-step or 2 types of interactions between the 2 proteins, N interacting with P to form droplets, and then further recruitment of N to the existing NP droplets. This suggests that P forms a dynamic scaffold to which additional N can be recruited. And to an extent, this goes along in a very hand-waving way, I agree, but it goes along with the fact that FRAP of P is slower than N in these liquid droplets, and therefore it may be some kind of scaffolding, dynamic scaffolding protein. Finally, if we add RNA, of course, there's a lot of talk of droplets, these kind of droplets being viral factories and having some kind of function. Of course, they have some kind of function. We wanted to know whether if we added RNA to our mixture of N and P inside the droplet, whether this would then form nucleocapsids. First thing that we saw is that RNA indeed co-localizes. So fluorescein labeled RNA here localizes very rapidly and is nowhere else in the sample. And within about an hour of incubation, we see nucleocapsids formed by electron microscopy, and also using the assay that we discussed previously, which is looking at the P peptide leaving the complex, we can see that in fact the formation of the nucleocapsids is accelerated in the droplets, which is more or less what you would expect, because of course the concentration of these 2 proteins in the droplet is very high. Okay, so that's all I'm going to say about measles now, and things will become much more random because I've never presented the next part before. This was published, it is actually published, it was published 2 days ago. But there haven't been many conferences and there hasn't been much opportunity to present it. What I'm talking to you now about is influenza. So as I'm sure many of you are aware, and a lot of the protocols that we're putting in place now for COVID-19 were actually developed for precisely this problem. If avian influenza adapts to humans, then this causes the possibility of a major pandemic, which could be extremely dangerous for human health. And in fact, the adaptive mutations are present in 2 proteins principally, and one of these proteins is the polymerase, the influenza, the RNA-dependent polymerase. They're concentrated in the PB2 subunit of the polymerase, and they're concentrated at the C-terminal end of this subunit. Because of this, now these are the mutations which need to occur in order for avian influenza to infect humans. Because of this, there's been a great deal of work in the past and in the last 15, 20 years to study this region, in particular this region, which is a two-domain region, a 627 region, a 627 domain and an NLS domain, which when crystallized actually pack very tightly together and form a very closely packed structure. And the avian and human adapted forms of these two proteins when crystallized are essentially identical. So structural biology doesn't tell us exactly why these mutations and what these mutations are doing, except that they're on the surface. And I should mention in particular one mutant, which is E627K. This is why this domain is called 627. This is the molecular signature of most influenza pandemics, actually all except one, and we think we know why. The other one is actually an exception. Okay, but when you study, we studied this a number of years ago, when you study this protein in solution, actually it doesn't look like all the crystal structures of all of the different mutants, but in fact, this is indeed present in solution, but it's in slow exchange with an open form of exactly the same protein. We know it's in slow exchange, it gives us two peaks, one peak reporting on the crystal structure and one peak reporting on the fast exchange average of this ensemble, which is then delocalized. So they are no longer, if you like, there's actually a salt bridge which keeps them together, and this is actually broken in the open form. And this occurs at 10s or 20 times per second at room temperature. And I should say there's also an NLS peptide at the end of this, these two domains, which is used to import the PB2 subunit into the nucleus. We studied this. I won't talk about this part of the study. I'll just summarize it very quickly. Using NMR, SACs, and single molecule FRET, we got a very concordant and beautiful description of this conformational equilibrium between open and closed forms, and we could show that only the open form binds importing to enter the nucleus. Approximately the same time, then structures started to come out of the full-length influenza polymerase with the three subunits shown here in yellow, green, and pink. And it was shown by our colleague Stephen Cusack. He determined the structure, the first structure in Grenoble. He determined the first structure of the viral RNA bound polymerase, and there these two 627 NLS domains have the same structure as when crystallized. But in another study, a couple of years later, then they could see that when the RNA was no longer there, then again, these domain dynamics start coming into play. And the 627 domain is here, bound to PB2, and the NLS domain is over here, bound to PA. So this alternative conformations in reconstituted polymerases, this clearly happens not only in the isolated 627 NLS, but also in the reconstituted polymerase. And most recently, a major step forward in the understanding of influenza adaptation, or avian influenza adaptation to humans, was made when Wendy Barkley and colleagues, and this was in 2016, identified a host factor which is responsible for this adaptation signature. This is actually ANP32A. It's a transcription factor. It has a folded domain of about 150 amino acids and an unfolded domain of either 100 amino acids or 130 amino acids, depending on whether this is in human or the avian form. The avian form has an insert, which is essentially a repeat of the preceding 28 amino acids and then an additional hexapeptide. And this is then 32 amino acids longer. When this is present, then the avian polymerase 627E works. When this is present, it requires 627 to be a K. And this is there's some kind of concerted cooperative adaptation that the way that the virus has found to infect humans with, in the presence of this shorter form, is to make this K mutation. So we wanted to investigate what the molecular basis of this is and how this one mutation can compensate for a 30 amino acid insertion. So we studied the interaction. This is the interaction of ANP32A with 627 NLS. I should mention this is a highly repetitive sequence. It's 60 or 80% aspartic acid or glutamic acid. And Aldo Camacho-Zarco, who is the first author of the article which has just been published, he did a massive amount of work to try and assign as many of these as possible. As you can see, there is indeed an interaction. This is with the human forms and the human adapted forms. Here we're looking at relaxation measured at different titration admixtures. And what you can see here is that the interaction is actually between 627 NLS and ANP32 is distributed all along this long intrinsically disordered sequence. So there seems to be some kind of multivalency or polyvalency going on. These are relaxation measurements and chemical shift titrations of the full length protein. This is the folded region, this is the unfolded region. You see essentially the same things, but you can see that the folded domain is also involved in the interaction. The interaction is distributed along the chain. The avian unique hexapeptide gives the largest chemical shifts in the avian-avian pair. The cross interaction gives very small chemical shifts. That means between human and avian adapted forms. The chemical shifts in the avian-avian interaction depend on the presence of the structure of 627 NLS. That means that when you have only 627 or NLS, there are very, very small chemical shifts. And crucially, the affinity is very, very weak in all cases. So it's between 800 and 1500 micromolar. Okay, so what happens when we look at it from the other side of the interaction? This is from the side of 627 NLS, in this case 627 NLS K. That will be the human adapted form with human ANP32A. This is the distribution of the chemical shifts. You can see them here. I've mapped them onto the surface of the two domains, of this representation of the two domains. It's a basic protein, 627, and therefore you can see that, of course, there are lysines and arginines on the surface, but only one of these two surfaces that contain these positive charges actually seems to be involved in the interaction. So there's some kind of specificity involved here. If we look at the same map using the avian adapted forms, and we just look at those additional shifts which occur for the human pair, you can see that they're all located in this surface here, which is actually the surface around 627, around 590. These are precisely the amino acids which are known to mutate in pandemic forms of the polymerase. So this is more or less summarized here. I should just say one more thing, and that is that this equilibrium, which is around 50-50 at room temperature, if we add AMP32, then the equilibrium shifts almost entirely to the open form in avian. There's a little bit left of the closed form in the human pair, but in the avian form, the open form is basically the only form that is present. And one last thing I should say, which is very important, and in this case, the affinity, if we observe from the side of 627 NLS, the affinity is approximately 20 micromolar. It's still approximately 600 micromolar if we observe for the E, for the avian form. Okay. Now, because we're in the presence of a very weak complex, which is highly dynamic, we chose the following approach, which was to use paramagnetic relaxation enhancement, intermolecular paramagnetic relaxation enhancement. So we have 8 different cysteine mutants, and these 8 different cysteine mutants, 5 of them are in the 627 domain and 3 are in the NLS, or one of them is more or less on the edge of the linker. What you'll see immediately when you look at these are the PREs, these are intensity ratios, you'll see that there's some kind of structure here in the folded domain. This comes from the repetitive nature of the folded domain here. And so this allows us, I won't go into the details, once you've done the correct averaging of the PREs over the different side chain conformations and taking into account the affinity and how much of the BAM form is present, we can determine the optimum pose of the 2 folded domains. And we can see that they attempt to be stabilized. They seem to be stabilized by hydrophobic and electrostatic interactions in this interface. How do we deal with the unfolded and disordered domains in such a dynamic complex? Well, Christian alluded to this. So we do extensive amino acid specific statistical coil sampling of ANP32 IDR of the unfolded domains and of 627 NLS linker and the peptide regions at the NLS, NLS C terminus. This is a lot of disorder, of course. And of course, we use coarse grain steric exclusion, and we calculate, so these would basically look like this if we calculated 3 of them, but in fact, we have to calculate many tens of thousands, or 100,000 of these. We calculate the paramagnetic relaxation for all of these, and then we calculate the optimum. We select the optimum ensembles using the asteroids algorithm on the basis of agreement with the experimental intermolecular NUI. So this is the agreement of the experimental data. So on the left, you have the human-human pair, and on the right, you have the avian-avian pair. What I mean is avian-adapted polymerase with avian AMP32 and a human-adapted polymerase with human AMP32. I won't go into the details too much now. There are clearly differences that you can see. The PREs again support polyvalency because there's broadening over 50 amino acids, as you can see here. but actually over here there is broadening, maybe less broadening, but it's even over a longer region, maybe 100 amino acids. The most efficient relaxation is by probes on the proximal interface, the same that was identified as being differential chemical shifts between these two complexes. So this all seems to be consistent. There's more contact with the NLS domain in the avian-avian pair. But this doesn't help very much looking at these two ensembles. You can look at these till the cows come home and you won't learn anything. You have to calculate something from them and see how they are different. And this is what we did. So we calculated a difference distance matrix. This is an average matrix. It basically tells us about the proximity of different conformers to different regions of the protein. So here we have the entire 627 NLS and the first 100 amino acids of the disordered domain of ANP32. And what you can see is very, very clear. There are closer contacts, all populations, of the 590, 627, 630 surface with the initial 40 amino acids of ANP32 in the human-human pair, whereas the closer contacts are rather between the linker and the NLS regions up here in the avian pair. Now, this first observation here, so this means there are many, many conformations which are around this surface, this basic surface. This substantiates the observed polyvalency and the avidity. Remember, in order for the avidity, in order to have an affinity which measures 20 micromolar on the surface of 627 and 600 micromolar on any given site on the surface here, you're going to have a lot of avidity going on in order for this polyvalency to have such an effect. And this is precisely what we're looking at, and it's what the PREs are telling us as well. Okay, so it's a good idea. I should probably get on with this because I'm probably going on far too long. We need to look at the cross complex, of course. This is what shouldn't work. This is non-adaptive polymerase to human HNP32A. Suffice to say, the interactions which are stronger here are weaker in the cross complex. There are fewer and weaker contacts in the regions where the adapted molecular mechanisms appear to be present. So this makes sense as well. Finally, of course, we need to know whether this interaction actually occurs in the presence of full-length polymerase. So our colleague Stephen Cusack gave us his polymerase, some polymerase to work with. And we looked at the interaction. You can probably recognize this signature. It looks very, very similar to what we looked at earlier. This is 4 micromolar of of ANP32A with 32 micromolar of influenza polymerase. The chemical shifts are very similar. We therefore decided to take the next step, which is to superimpose 627 from our complex onto the 627 of the crystal structure of this sample. And basically, you can see, I don't know if you can see, but the ANP domain actually slots very nicely into a cylindrical pocket, which is formed between PB2 and PB1. And in fact, there are 2 recently identified adaptive mutants, which are on the surface of PB2 here, for which no reason could previously be found, but now we can see that in fact ANP32 is right next to these 2 mutants. And one can speculate about the sampling radius of the intrinsically disordered domain and what it might be useful for. It may play a role in dimerization of the polymerase, which has been muted to be important for replication. It may regulate the interaction with RNA polymerase II to promote transcription and replication. This is another possibility. We don't really know this yet. I should just mention the more open form of the polymerase. Of course, the ANP32 can be easily docked in the same conformation as we've determined onto 627. So either of these 2 different conformations of polymerase are compatible with our results. Okay, so just to conclude, this is the final slide. So comparison of human and avian adapted 627 NLS domains of influenza polymerase in interaction with a host transcription factor ANP32A identifies key differences. The highly charged acidic intrinsically disordered domain interacts transiently with the basic face of 627. In the human adapted case, this is polyvalent, exploiting the electrostatic interactions that are enhanced by the crucial 627K mutation. When this is not present, and when 627 is an E, this basic surface is interrupted and this polyvalency is no longer visible by NMR. The cross interaction exhibits neither of these stabilization mechanisms, which may make sense. The interaction is maintained in full-length polymerase, and therefore we can speculate. And in fact, we can use this molecular framework to start answering questions as to what the intrinsically disordered domain is actually doing in this crucial complex. Okay, so I've probably gone wildly over, I'm very sorry. I'd like to thank the people who did this work. Now, Aldo Camacho-Zarco, he came to our lab, I don't know, 3 or 4 years ago from Markus's lab. He's done an amazing amount of work, and you can probably imagine this is quite a complex to work with, and he's really done a brilliant job. And I'm sure this work will be very important for understanding the function of what is going on here. Serafima Guzeva, she did all of the work on the measles droplets, and Sigrid Millers did all of the work on the measles complex, which I discussed with you. I'd like to thank you for your attention. I'd like to thank my collaborators, Rob Rugok, Stephen Cusack, and Darren Hart, and Guy Schoen, and Denis Gélier. And I'd like to thank you for your attention. And I'd love to answer questions.
Yeah, thank you very much, Martin. Thank you very much also for staying quite in time.
Oh, I did. I had no idea.
And not talking too fast either. And maybe I'll just start with the Q&A here. Actually, Walter Chasin had the same question as me. The 5 order of magnitude weaker interaction site in the MEF long tail is for a binary measurement. There is a higher local concentration due to linkage to the stronger site than in the binary interaction. Do you have a sense or made a calculation of the effective KD, the weak interaction in the context of the full length construct?
Yeah. Well, unfortunately, when you do that, of course, we've done that. Thank you, Walter. That's a very good point. If you think about rotatable bonds and George Whiteside's calculations, we have a lot of rotatable bonds between in 150 amino acids. So essentially,
maybe I could follow up on this question. I mean, if you cut the N terminus from the rest, I mean, then in principle, this effect of increasing local concentration would be smaller, and then if all the components are there, but the N terminus is cut from the rest, including the Hell domain, is it then still?
Oh, but this is then I didn't explain myself. I probably spoke too fast. So we determine the 600 micromolar in the absence of the N-terminal domain, of course. Otherwise, we can't determine it for that reason.
Yeah, sure.
So the 600 micromolar is the intrinsic KD in the presence of the KD of the main interaction. Of course, it's different. How different it is, this is ongoing work. We're looking at that. We're also making the linker shorter. You have to make it a lot shorter before it starts making a significant difference, is my feeling. At least if you think about this in pure physical chemistry terms, or the way things have been studied using physical chemistry. However, I think as well that there are other interactions along that sequence between residue 50 and residue 180, which mean that there are other cooperative effects which may make the calculation a little bit more complicated. But yeah. These are all very interesting questions. I mean, I agree. This is we are investigating this, but the intrinsic KD is 600 micromolar. That was measured in the absence of the main interaction site. Otherwise, we can't determine that.
Okay, so Walter is not writing anything in order to follow up. At least I don't see. No. So then we go to Jay Deep's question. First of all, he congratulates you for the very nice talk, and second, he asked, did you any possible interaction that drives the phase separation in presence of RNA?
Yeah, now that's a very interesting question. It's a very good question. So first thing I should say, if we're looking for cation pi interactions, which are often evoked for stabilizing droplets, we actually don't have really that many of the right amino acids in order for this to be stabilizing our interaction, our droplets. But then I think this is probably just that there's lots of the alphabet that we don't know yet about weak interactions that can stabilize droplets. And I do believe that once you add RNA, then you will anyway disrupt whatever was stabilizing the interactions between the two proteins will then become very different once the RNA enters the droplets. And anecdotally, they do look a little bit different. Now, we haven't made enough measurements. We'd like, of course, to be able to stabilize these droplets enough so that we can do NMR measurements of what's inside them. And then we can see how that is modulated by RNA. We're developing other model systems to do this currently. And so this is ongoing work, but I haven't got any answers for you yet.
Okay, the next question is, what's the minimum number of confirmations that would be needed to fit the PRE data? How many confirmations do you have in your ensemble?
there are 60 conformations in the ensemble. How many would you need to explain the experimental data in the disordered domain? Well, we optimize that number. This is basically by back calculation. We try and determine where the chi-squared of the data which are left out is in a very broad minimum, and that's around between 60 and 80 conformers. And so therefore that implies that anything less than that is not reproducing the experimental data for obvious reasons. I think if you look at the interaction profiles, it's going to be very, very difficult to have 100 amino acids in sufficiently close proximity to one PRE probe in order to provide such a broad-based, broad-bottomed potential along primary sequence. So I think even logically, you can think, well, one is clearly not going to do it. But 60 for the moment is the optimum number. Now, if we had more data, that would maybe be a different number. That's, I'm fully aware.
Did you say 60, 60, 1?
60.
60, okay. The next question is, please could you tell me why the KD of 200 for a KD of around 600 micromolar, can one get a lower KD with another technique? I must say I don't understand the question. Martin, maybe you can.
I missed a bit of it in the middle. Can I explain how?
So could you tell me why the KD of 200, I guess it's KD because, yeah, it's KD, for a KD of around 600 micromolar, can one get a lower KD with another technique?
No.
Do you understand, or shall we?
I don't understand the question, actually.
Yeah, me neither.
Maybe we can...
Mark Sanderson has raised his hand, so could anybody make him speak? Or maybe while this is done, we go to Catherine Royer. Great talk, Martin. I recognize my old friend PP32.
Yes.
The C-terminus of the PP32 folded domain that interacts with polymerase, question mark?
Yes, so that's the question. Yes, Kathy, indeed. I've been reading all of your articles on PP32. So ANP32A used to be called PP32, so I should have mentioned this. And Kathy did some beautiful work using high pressure NMR to measure the stability of these different repeat regions. As far as I remember, Kathy, I think the most stable region is the C-terminal region, in fact. My answer to your question is yes, the C-terminus does interact. And there are some quite important mutants which have been shown. AMP32A, AMP32B in some species are interchangeable. There are differences in this region which have been evoked to be important. There was an eLife article recently which suggested this. And where these differences are is exactly where we see the interaction. So the interaction between the two folded domains, at least. So I do believe there's a lot of work to be done, and probably a lot of work to be done on stability. It has to be said that most of the pull-down assays that are providing virtually all of the experimental data, except the NMR that you've just seen, are not really doing tests on whether these mutations are changing the stability. And I think it would be good to go back to a lot of your work, Kathy, and try to check to see if any of the mutations are actually being made are mutations you've already made. Could also be. And then we could learn something about the stability of the protein.
So Kathy writes, the C-terminus is most stable.
Right, that's what I thought.
Now I think the anonymous is probably Mark Sanderson because he is now entered this talk.
Yeah, so I thought it was a very nice talk, Martin.
Thank you.
Now my question was why the ESD is 200 for a KD of 600. That's quite a large ESD. And is it possible to get a lower ESD, standard deviation using some other technique?
Sorry, yes, okay, this is from relaxation dispersion, yes, okay, yeah. So we make a lot of suppositions when we measure this, and you're right, 600 plus or minus 200. Of course, by other techniques, I hope you'll be able to measure that more precisely, but yeah, I do admit that this is, we measure relaxation dispersion at 2 fields. If it's a relaxation dispersion, the eagle-eyed of you will say, well, that's a pretty crummy data, Martin, but they're what we have, and they make sense within the error bars that we cite. But I agree, I'm sure this KD can be measured more precisely.
And my other question is, what is known about the packaging within measles of the RNA?
Of the RNA? You mean within the nucleocapsid?
Yeah.
What do you want to know? Quite a lot. So it's known how it binds. Now we know a bit more because of the structure we just determined. Now we can see that really how it binds. There are 3 bases that bind into the cleft, and 3 bases which point ostensibly out of the cleft, but they're still stabilized by protein interactions. We know from our study that the register is always maintained. There are no holes, and therefore we can see that at the end, the last 3 nucleotides are actually accessible. This is a 3' end, and that this may have implications for accessibility to the polymerase. This, again, we didn't know until we determined the structure. What else do you want to know? Is there anything specific about?
No, it's just like within there's a lot of, before infection, is there a lot of fluidity of the RNA within the capsid? Is it?
Oh, you mean, not to my knowledge. I think
right.
Once it's bound in the way it's bound. Now, of course, at some point, for accessibility to the polymerase is really not understood. And this has something to do with both the phosphoprotein and the nucleoprotein. And we'd like to be able to study that, but so far it's kind of in the realm of speculation until we can reconstitute all of those elements and look to see really where the polymerase is with respect to the nucleocapsid. It's going to be a little bit difficult, but obviously there needs to be some fluidity because otherwise there'd be no replication or transcription, but yeah.
No, sure. Yeah, okay. Thank you much indeed.
Thanks.
So we have another 2 questions, I think, before we go to the more informal chat after this. And the first question is, do you have any idea of the interaction stoichiometry? Now, I'm not exactly sure
which interaction. Hold on.
Maybe you answer for both parts of the talk.
Oh, sorry. No, sorry. I can. What did you say, Christian? What was the last bit?
The interaction stoichiometry. Whether you have any idea about the interaction stoichiometry?
Okay. So I'll go. I mean, I think I know which one because it's actually a very interesting question. I didn't mention it, and I thought I hope somebody picks up on that because I didn't have time to mention it. So I hope you're talking about the stoichiometry between the 627 NLS, the human adapted 627 NLS, with the human AMP32A, because this is where the affinity suddenly shoots up, because what we think of is multivalency, and there indeed we have evidence that the stoichiometry is at least 4, if not between 4 and 8 to 1. So it's really very, very high. And the methods that we've used to interpret, I mean, we have pretty, I don't think our data is up to an analysis which will go in any more detail than that, but it's certainly higher than 1 and makes sense in terms of multivalency.
Okay, and actually this question was by Lu Yun Lian, and the last Ho Li. Thank you for a great talk looking at the wide spectrum of your IDP studies. I'm just curious about how you pick up these interesting systems.
We got hundreds in the fridge that we just throw away with them. I'm not measuring on that. No, that's not true, really. That's just the way it is. I mean, that's just how it happened. Did I understand the question correctly? Do we throw away a lot of systems that don't give nice spectra? Is that the implication? Believe me, we have some pretty crummy spectra as well. I mean, you could argue AMP32 is. We've talked to Aldo about it. It's pretty crummy. You know? It's about 50% aspartic acid, and he spent a lot of time assigning what he could, and you know, it's not easy. So now we just deal with what we get, Louis Young. You know what it is. But when we get a nice one, we show it as many times in the talk as possible. Maybe that's it.
Okay, we have another one from Fito, and since he worked with you for some time, so I mean, I think I cannot kick him out. So thanks for the great talk, Martin. You mentioned you can measure HSQC spectra in phase separation conditions and calculate the amount of protein on each phase. Do you have an estimate of the exchange rate of proteins between the 2 phases? Yeah,
this is assuming that there's no exchange affecting that measurement. So that's assuming it's fast, which we don't know. So yeah, absolutely.
You see only one set of signals.
Yeah, exactly. Yeah, good point, Fito.